Hello student, we continue the topic Cell the Unit of Life class 11th lesson with topic Golgi complex. It is discovered by Indian scientist Camillo Golgi in 1898 from nerve cell of cat. This is the important term most of the time asked in NEET examination. Golgi complex is found in all eukaryotic cells, means it is absent in prokaryotic cell except human RBCs. It is also known as lipochondria, idiosomes, Dalton complex and Golgi apparatus. Rather than Golgi complex, so many terms are used for this one. Distribution. They are found in higher plants. Cell in scatter form. In higher plants like angiosperm, gymnosperm, pteridophytes, they are present in the cell but not organized. They are scattered all over the cell. And 10 to 12 number per cell in case of plant cell. In animal cell, they are localized with specific structure, specific arrangement and found in 3 to 7 per cells. Golgi bodies are not found in prokaryotic cell. We already talked about that. They are present in eukaryotic cells and absent in prokaryotic cell. Origin. Basically, Golgi bodies are originated from endoplasmic reticulum. In last video, we talked about the endoplasmic reticulum. Now we see the structure of Golgi bodies or Golgi complex. It is double membranous unit, shows 3 compartments in structure: cisternae, vesicles and vacuoles. It is, now firstly we see the cisternae. It is elongated, tubular, flattened, sac-like part filled with fluid. These bundles are parallel to each other and these are double membrane unit and the thickness of the membrane is 60 angstrom. Now we try to draw here cisternae. That is the nuclear membrane of the part, cell nuclear membrane, and near to the cell nuclear membrane the endoplasmic reticulum is attached to the membrane. It is located in this way that rough endoplasmic reticulum near to the nuclear membrane. We already talked about rough endoplasmic reticulum in last video. All over they attached to the that cisternae part. Cisternae are arranged in this way that they forming like structure. This is cisternae. Now the next one is vesicles. These are a bulbous outgrowth arranged in periphery. It stores the secretory materials. The diameter of vesicles is 60 mu. Now here in this page we start to draw the vesicles. These are double layered structure, we talk about it, and the thickness is 60 angstrom, double layer structure. These are also sac like, means they basically look like that, this sac like structure. And near to it the vesicles, the small part is attached, we call vesicles. They are secretory in function. The third part is vacuoles. These are large, rounded, sac-like structure found at the edges of Golgi complex. Now here we again draw the structure of vacuoles. At the edges of the Golgi complex, their large secretory structure is present, and that large secretory structure we called vacuoles. The Golgi body has two phases. The first phase we called forming phase, it also called proximal phase, and it also called cis phase. If it is towards the nuclear membrane, means if the phase is towards the nuclear membrane and endoplasmic reticulum, is called forming phase, proximal phase or cis phase, and its shape is convex. Now we see in our diagram that this phase is called the forming phase because it is towards the nuclear membrane and it is towards the endoplasmic reticulum. The location is towards them, that's why it is called forming phase or proximal or cis phase. And if it is towards the cell membrane, here the cell membrane is present, that we call the plasma membrane. It is the outer covering of the cell, so it is cell membrane. And if the face of the Golgi complex towards the cell membrane, then that structure we called trans phase. That phase we called trans. It is also called maturation phase, distal phase or trans phase because it is present towards the cell membrane, opposite to the nuclear membrane, and it is concave in shape. The cis phase is convex in shape while the trans phase is concave in shape. Now we move towards the function of Golgi complex. The main function of Golgi body is secretion. It secretes a lot of enzymes, hormones. Lysosomes are formed from Golgi bodies. Lysosome का development, formation from the Golgi bodies it takes place. It helps in the synthesis of large molecules of complex carbohydrate. Synthesis of large molecules of complex carbohydrate means the small units or small subunits of the carbohydrate combined to form a single complex molecules of carbohydrate. It helps in formation of acrosomes in sperm and matrix of cartilage. Cartilage is the part of our endoskeleton, and this is the structure of sperm. It is look like that. This is the tail part, and the upper part of the sperm that we called acrosome, which contain the enzymes which are responsible to crack the zona pellucida in female egg, and that acrosome part is made up of Golgi complex. It helps in cell plate formation. Basically, cell plate formation takes place when the cell divided into two cells, means by the process of mitosis. During that process, after cytokinesis, the cell is divided into two types and two parts, and before that the cell plate formation takes place between that two cells. Plant secretory products like gum, wax, mucilage are released by these body. Latex, gums, wax, these are the secretory products of the plant cell, and all these are released with the help of Golgi complex. Secretory cell secretes hormones, enzymes, mucus and albumin, etc. In case of animal cells, the secretory cells of the Golgi complex, which are the secretory cells? Vesicles. These secretes the hormones, enzymes responsible for the lysis of the cell matter, means these are help to formation of the lysosomes. So It also prepared the enzymes of lysosomes, mucus and albumin in egg. These are the some secretory functions of the Golgi complex. This is about the Golgi complex. If you want the notes about that topic, then you have to check the description box. Thank you.